Hello and good evening to everyone. Today we are going to discuss the last assignment, which is the DNA replication question. My name is ANATI NABILAH, BINTI TAJUDIN SHAH, and my machine number is 205706.
My name is ZARIFAH, BINTI AZAMUDDIN. My machine number is 207809.
My name is NURHAN bin MAHADZIR. My matric number is 208241.
Hello everyone, my name is Timothy Lee Zhou and my matric number is 207980.
Good afternoon, everyone. My name is GUAN. My matrix number is 206974.
Hi everyone, my name is NUREEN AFISHA ADNAN and my matrix number is 208291.
So today we're gonna answer this question. So the question state that illustrate the replication of the following DNA strand end to end. So from this question, we know that we're gonna illustrate the whole process of DNA replication. We know that the DNA replication basically divide into 3 steps, which is initiation, elongation, and termination. So let's look deeper into the question. And we have, based on the information given, we have only 2 DNA strands and 2 origin of replication. So based on the information, we know that this is a eukaryotic DNA because has 2 origin. And for if the DNA is prokaryotic, which means it has only 1 origin. So let me draw something. So from here we know that the DNA will actually start to initiate, and it will become something like this. Right. So from here we know that if this DNA strand start to replicate, it will produce this kind of figure, and it will has actually 4 replication fork. So based on this information, we don't have much information about this DNA strand, but we know that if the DNA strand start to replicate, it need to methylate, which means that it will add something like, something like Ne to indicate the DNA strand are actually mature. So it will prevent the DNA to mutate after the many replication. Yeah. So although we don't have this kind of information, but we still can discuss about how it actually start to initiate. But how? We know that the DNA strand are actually in a supercoiled state, and beside that, it has also, got many forces to stabilize the structure, such as the hydrogen bond between the adjacent DNA strand. So, Shade, do you mind to share out how the initiation occur to overcome this problem?
Okay, so as like you said before, that DNA is maintained in supercoiled state, but this is a problem. We call it as topological problem. So when in DNA is maintained in supercoiled state, but DNA replication requires the DNA, the 2-strand DNA, to be separated. So we have to find the solution. So there is topoisomerase as the solution for the topological problem. So what is topological DNA topoisomerase? DNA topoisomerase is an enzyme that catalyzes breakage and union reaction, which is releasing torsional stress ahead of replication fault. So DNA topoisomerase got two types, which is type 1 and type 2. For type 1, they break a single stranded DNA strand, which is release one strand and pass the second strand to the gap made, then seal the nick. The second one is break the both strand of the double helix and create gate through which a second segment of the helix is passed. So there is mechanism for the topoisomerase 1, which is type 1 for the type 1. All the topoisomerase cleave DNA using a covalent tyrosine DNA intermediate. This is because the relation of DNA supercoil is energetically favorable. So the reaction proceed without using ATP. So let me explain for the initiation. First is formation of replication fork. So the both strand of DNA after they separate, before they separate, become replication fork. The name why they call as replication fork is because the junction between the newly separated template strand and the unreplicated RNA look like this, look like they divide into two, so they call it as a fork. So many protein involved in the initiation of replication. Replication fork is initiated at the origin of replication, which prokaryotic organism have only one ORC, whereas eukaryotic organism have many. So linear chromosome of eukaryotic organism have many origin of replication. Two replication fork are initiated at the origin of replication, which is one initiate. Two replication fork emerge from the duplex and progress in opposite direction. There is only one origin of replication is available, is in a circular chromosome. So why only fully methylated origins of replication can initiate a replication? So DNA must be methylated to initiate replication. DNA must be methylated to initiate replication. So there is specific features of origin replication, which is characterized by unique DNA motif. So you must thinking, what is DNA motif? DNA motif is a nucleotide sequence pattern that serves some biological function. In DNA replication, it refers to the component of the origin of replication. So that is DNA motif. And then the initiation, I will explain about the initiation of replication at RDC. DNA a protein bind to the 9BP motif. So this is binding require ATP. The 380 reach 13 repeat melting, which is denature two strand separating, giving way for DNA B, which is helicase, to claim the opening strand. So the DNA B protein bind to each strand, which help from DNA C, which is helicase. Activity of DNA B hydrolyzes H bond between the two strands. So you must wonder, what is DNA A, DNA B, and DNA C? So the function of DNA A is recognizes ORI sequence, which is open duplex at specific site in origin. Meanwhile, DNA B, which is helicase, is helping to unwind the DNA to become separated. And then for what is DNA C is required for DNA B binding at origin. So Tim, what do you think about SSB, which is single stranded binding protein?
Okay, thank you, Syed. So SSB protein, which is single strand bind, once separated, a SSB binds to the single-stranded DNA strands. So once binding, it promotes the binding of another SSB to the immediate adjacent ssDNA, which is what we call cooperative binding. The purpose of this binding is to prevent the ssDNA strands from being self-annealed and from being hydrolyzed by nuclease. There's another purpose of this binding is to prevent water from base interactions. In the initial stage, it requires an RNA primer. The DNA polymerase cannot initiate DNA replication on its own, so it requires a short primer. I'm going to give you the definition of RNA primer first. RNA primer is an RNA which initiate DNA synthesis. The primers are required for DNA synthesis because there is no known DNA polymerase is able to initiate polynucleotide synthesis. DNA polymerase are specialized for elongating polynucleotide change from their available 3-hydroxyl terminate. In contrast, RNA polymerase can elongate and initiate polynucleotides also. Primers are special RNA polymerase that synthesis short-lived oligonucleotides used only during DNA replication. Even though transcriptional RNA polymerases primary synthesis messenger RNA, transcripts are sometimes used to initiate DNA synthesis. For instance, the single-stranded DNA phage M13 genome utilizes RNA polymerase instead of primase to initiate its DNA synthesis. In addition, the dominant hypothesis concerning mitochondrial DNA replication initiation is that the mitochondrial RNA polymerase synthesizes a polymer that is not displaced from the template. Then the special RNase MRP cleaves the ribopolymer at the specific size, enabling the exposed 3-hydroxyl termini to serve as primers for DNA synthesis. Finally, transfer RNAs make up a special class of primer RNA because certain species of tRNA are used by retrovirus reverse transcriptase to initiate replication of retroviral genomes. It is also possible to initiate DNA synthesis without primer RNA. The initial proteins of adenovirus and are attached to both of the 5' end of linear duplex DNA and provide a serine beta hydroxy group from which a DNA polymerase elongates. Another example is that many plasmids encode sequence-specific nuclease which cleave one strand of the duplex to create a 3' hydroxyl for the host DNA polymerase. An example of an animal virus is parvovirus, where the 3' end of the parental strands form a DNA hairpin and becomes a primer of its complement. And for the RNA polymerase, which is also called as primase, RNA polymerase are also known as primase, which is an enzyme that synthesizes RNA primers. Special proteins are responsible for leading primase at the RIC so that leading strand can commence. In a subsequent step, other replication proteins cause primase to initiate DNA replication on the opposite lagging strand. After both the leading and lagging strand primers have been elongated by DNA polymerases, the RNA primers are enzymatically eliminated, resulting a gap which is filled by DNA polymerase 1 and DNA ligase. So now I'm going to talk about the elongation. During the elongation, an enzyme called DNA polymerase adds DNA nucleotides to the 3' end of the newly synthesized polynucleotide strand. The template strand specifies which of the 4 DNA nucleotides, A, T, C, or G, is added at each position along the new chain. Only the nucleotide complementary to the template nucleotide at that position is added to the new strand. DNA polymerase contains a groove that allows it to bind to a single strand template DNA and travel one nucleotide at a time. For example, when DNA polymerase meets an adenosine nucleotide on the template strand, it adds a thymidine to the 3' end of the newly synthesized strand, and then moves to the next nucleotide on the template strand. This process will continue until the DNA polymerase reaches the end of the template strand. DNA polymerase cannot initiate new strand synthesis. It only adds new nucleotides at the 3' end of an existing strand. All newly synthesized polynucleotide strands must be initiated by a specialized RNA polymerase called primase. Primase initiates polynucleotide synthesis and by creating a short RNA polynucleotide strand complementary to template DNA strand. This short stretch of the RNA nucleotides is called the primer. Once RNA primer has been synthesized as the template DNA, primase exists, and DNA polymerase extends the new strand with nucleotides complementary to the template DNA. And eventually, the RNA nucleotides in the primer are removed and replaced with DNA nucleotides. Yes.
Tim, thank you for your explanation. Yeah, so just now you basically explain some of the initiation, like the SSB protein bind to the replication fork to serve, and it has some serve some function, and then you got explain a little bit of the primer, and then yeah. So let's move on to REPLICATION and ANATI. Do you have any things to add on? What happened during the leading strand and lagging strand? Yeah, thank you.
Okay, so for the leading and lagging strands, since DNA polymerase, it can only synthesize new strands in the 5' to the 3' direction. Therefore, two newly synthesized strands go in the opposite direction because the template strand at each replication fork is anti-parallel. The leading strand is synthesized continuously towards the replication fork, as the helicase would unwind the template double-stranded DNA. For the lagging strand, it is synthesized discontinuously in the direction away from the replication fork and also away from where the DNA helicase unwinds. The lagging strand is synthesized in pieces because the DNA polymerase can only synthesize in the 5' to the 3' direction, and so it would constantly encounter the previously synthesized new strand. The pieces which synthesized by the lagging strands are called Okazaki fragments, and each of the fragment begins with its own RNA primer. And continue with REPLICATION.
Okay, since all know DNA polymerase can synthesize only in 5' to 3' direction, a problem arises in trying to replicate the two strands of DNA at the fork. So notice that the top strands must be discontinuously replicate in short stretches. Thus, the replication of both parent strands is a semi-discontinuous process. The strand that is continuously synthesized is called leading strand, while the strand that discontinuously synthesized is called lagging strand. So DNA primase is needed to lay down the RNA primer in a short sequence at the lagging strand. So later on, it will be removed by the exonuclease. This process is needed in this replication process so that the DNA polymerase could start replicate at the co-adding the DNA nucleotide to the to synthesize the DNA. So for the leading strand, it will only attach. It will only require one primer. But for the lagging strand, it will be multiple replication bubbles with 2 replication fork, each formed during the DNA synthesis. So we will continue to the next presenter, I think.
Okay, next is the next process is termination. At the termination, the replication forks meet and terminate at many points in the chromosome. And type 2 topoisomerases, topoisomerases are ATP-dependent enzymes, and it cut both strands of a DNA double helix. So there is a problem that could occur during the end replication. As the lagging strand replication machinery reaches the end of the chromosome, at some point, primase no longer has sufficient space to synthesize new RNA primer, and this will result to incomplete replication and a short ssDNA region at 3' end of lagging strand. So when this shortened DNA is replicated later, one of 2 products will be shortened and lack of region that was not fully copied. The shortened ssDNA can actually cause the loss of genes. So to prevent the loss of genes, the tips of eukaryotic chromosomes have these specialized DNA caps called telomeres. Telomeres consist of hundreds or thousands of repeats of the same short DNA sequence. The function of telomeres is it facilitates DNA replication in linear chromosome by forming caps to protect the DNA duplex from nucleases. So actually telomeres need to be protected from a cell's DNA repair system because telomeres have single-stranded overhangs that look like a damaged DNA. The overhang at the strand, some cells have the ability to reverse the telomere shortening by expressing telomerase. Telomerase is an enzyme that extends the telomeres of chromosome. So basically, telomerase is an RNA-dependent DNA polymerase, which means this enzyme can make DNA using RNA as a template. So how does telomerase work is by it extends or it adds nucleotides to the overhanging strand of the telomere DNA using this complementary RNA as a template. So when the overhang is long enough, a matching strand can be made by the normal DNA replication and producing double-stranding DNA. So the end replication problem that I mentioned before could be solved by the presence of active telomerase and the telomere. Okay, that's all for me.
So do you guys got any question to ask? If no, I want to ask some question. Yeah, so Tim, just now you mentioned about the SSB, single-stranded binding protein, that serve few purpose. One of the purpose is to prevent the water-based interaction. So why is very important, and actually how the water will interrupt during the DNA replication?
For this part, I search and I actually search and there's no answer to it. If you have any, I also want to ask about this also too. Do you have any answer?
In my opinion, because the DNA strand made out of many nucleotide bases, and each of the nucleotide bases has a functional group, maybe like OH or NH, and this kind of functional group can actually to have bond to produce a hydrogen bond with water, and I think that it will interrupt when the DNA replication are actually synthesis. When we added the nucleotide, the water will prevent it. So my answer is, yeah, it's very important for SSB to in there, so that it prevent the water to, you know, substitute the nucleotide bases to add in during the replication. Yeah. So that's my answer. So do you guys want to ask anything? One more things I want to mention about. I'm not sure that just now REPLICATION and ANATI got covered or not, because during the just now, like say that how to solve the 5' to 3' end of that kind of process to, you know, to replicate. Actually the DNA are very, you know, a lot of mechanism. One of the mechanism is the DNA are actually will form some kind of a loop, you know, form some kind of a loop like this, something like this. I'm not sure, but something like this. So for the synthesis towards the replication fork, this has no problem, you know. But for the lagging strand, actually it has produced some very specific structure, and it will let the strand, you know, to be synthesized into 5' end. So it kind of something like this. Do you guys got anything want to add on regarding this? So actually this kind of, you know, structure is actually is undergo local alignment, you know, and it will form a loop to represent, you know, 3' to 5' end template. So it's like the DNA play the, you know, play the direction. So at last it will also only synthesize through the 5' to 3' end. Yeah. So something like that.
Thank you for your explanation.
I think we have 1 more minute. Yeah. So I think that's it, right? You guys okay? Very nice. Very okay.
Okay.
Okay. Yeah. Okay. That's it for our group discussion on DNA replication. And thank you for watching. Have a nice day. Bye bye.
Thank you,
Bye.
everyone.
Okay. Team, remember